Meiosis. Meiosis क्या है? Meiosis is the production of offsprings. Bisexual reproduction includes the fusion of two gametes, each with a complete haploid set of chromosomes. Gametes are formed from specialized diploid cells. This specialized kind of self-division that reduces the chromosome number by half results in the production of haploid daughter cells. This kind of division is called meiosis. Meiosis encloses or it ensures the production of haploid phase in the life cycle of sexually reproducing organisms, whereas the fertilized restores the diploid phase. We come across meiosis during gametogenesis in plants and animals. This leads to the formation of haploid gametes. The key features are meiosis involves two sequential cycles of nuclear and cell division called meiosis 1 and meiosis 2, but only a single cycle of DNA replication. साइकिल जो होगी डीएनए रेप्लिकेशन की वो सिंगल होगी, लेकिन जो न्यूक्लियर और सेल डिवीज़न होगा वो दो बार होगा, एक मियोसिस वन में, दूसरा मियोसिस टू में. Meiosis 1 is initiated after the parental chromosome have been replicated to produce identical sister chromatids at the S phase. Meiosis 1 जो है initiated after the parental chromosomes have replicated to produce identical sister chromatids at the S phase. Meiosis involves pairing of homologous chromosomes and recombination between the non-sister chromatids of homologous chromosomes. Four haploid cells formed at the end of meiosis 2. Meiotic events can be grouped under the following phases: meiosis 1, meiosis 2. meiosis 1 में prophase 1, prophase 1, metaphase 1, anaphase 1, telophase 1. Prophase 2, metaphase 2, anaphase 2, telophase 2. meiosis 1 में जो है हमारा कौन सा आएगा पहले? meiosis 1 में आता है हमारा prophase 1. prophase 1 में prophase of the first meiotic division is typically longer and more complex when compared to the prophase of mitosis. जब माइटोसिस के प्रोफेज के साथ कंपेयर किया जाए तो ये प्रोफेज जो है मोर कॉम्प्लेक्स है. इट हैज़ बिन फर्दर सब डिवाइडेड इनटू फॉलोइंग 5 फेजेस. ये 5 फेजेस में फिर से डिवाइडेड है, और वो 5 फेजेस कौन से हमारे? वो 5 फेजेस हैं लेप्टोटीन, जाइगोटीन, पैकेटीन, डिप्लोटीन, डाइकाइनेसिस. लटकती जुल्फों पर दिल धड़का. ड्यूरिंग लेप्टोटीन chromosomes become gradually visible under the light microscope. The compaction of chromosomes continues throughout the leptotin. This is followed by second stage of prophase, one called zygote. During this, chromosomes start pairing together and the process of association is called synapses. Synapses are visible in zygote. Such paired chromosomes are called homologous chromosomes. Electron electron micrographs of this stage indicate the chromosome synapses is accompanied by the formation of complex structures called synaptonymal complex. Synaptonymal complex is also found in zygote. zygote में कौन सी मिलती है? एक तो हमारे homologous chromosomes मिलते हैं, synapses मिलता है, और क्या मिलता है? synaptonymal complex. The complex formed by pair of synapsed homologous chromosomes is called bivalent or tetrad. इसको क्या बोलते हैं? bivalent और tetrad. These are more clearly visible at the next stage. The first two stages of prophase are relatively short-lived as compared to the next stage. और नेक्स्ट स्टेज हमारी पैकेटीन है, तो पैकेटीन जो है ज़्यादा बड़ी है एज कम्पेयर टू लेप्टोटीन एंड zygote. During this, 4 chromatids of each bivalents become distinct and clearly appear as tetrads. This stage is characterized by the appearance of recombination nodules, the sites at which crossing over occurs between non-sister chromatids. of homologous chromosomes. Crossing over is the exchange of genes or genetic material between the two homologous chromosomes. Crossing over is also an enzyme mediated process, and the enzyme involved is called an recombinase. So, enzyme involved in the recombination is recombinase. Crossing over leads to recombination of genetic material on two chromosomes. Recombination between homologous chromosomes is completed by the end of packaging, leaving the chromosomes linked at the sites of crossing over. The beginning of diplotin is recognized by the dissolution of synaptonymal complex and the tendency of recombination homologous chromosome of the bivalents to separate from each other except at the site of crossing over. The X-shaped structures are called chiasmata. So chiasmata are formed in diplotin stage. In oocytes of some vertebrates, diplotin can last for months or years. Final stage of meiosis, prophase 1 is dikinesis. This is marked by the terminalization of chiasmata. During this phase, the chromosomes are fully condensed and the meiotic spindle is assembled to prepare for homologous chromosomes for separation. By the end of dikinesis, nucleus nucleolus disappears and the nuclear envelope also breaks down. Dikinesis prevents transition to metaphase. Metaphase 1. The bivalent chromosomes align on the equatorial plates. The macrotubules from the opposite poles of the spindle fiber attach to kinetochores of homologous chromosomes. Anaphase 1. Anaphase 1 में homologous chromosomes separate, while sister chromatid remains associated at their centromeres. Telophase 1. Nuclear membrane and nucleolus reappears, cytokinesis followed, and this is called dyad of cells. So dyad of cells is found in telophase 1. Although in many cases of chromosomes do undergo some dispersion, they do not reach the extremely extended state of the interphase nucleus. The stage between two meiotic divisions is called interkinesis and generally short-lived. Interkinesis is the stage between two meiotic divisions, which is very short-lived. There is no replication of DNA during interkinesis. Interkinesis is followed by prophase 2, much similar to prophase then prophase 1. जो प्रोफेज था माइटोसिस का उसके साथ सिमिलर है, बजाय कि इससे जो है लेस सिमिलर है। क्या है? लेस सिमिलर, इससे जो है मोर सिमिलर है। फिर हमारा आता है मियोसिस 2। मियोसिस 2 में हमारा प्रोफेज 2। मियोसिस 2 is initiated immediately after cytokinesis, usually before the chromosomes have fully elongated. In contrast to meiosis 1, meiosis 2 resembles normal mitosis. The nuclear membrane disappears by the end of prophase 2. Chromosomes again become compact, and in metaphase 2, chromosomes align themselves at the equator and microtubules form from the opposite poles to spindles get attached to kinetochores of sister chromatids. Anaphase 2. Anaphase 2 may it begins with simultaneous splitting of the centromere of each chromosome, which was holding the sister chromatids together, allowing them to move towards the opposite poles of the cell by shortening the microtubules attached to kinetochores. Telophase 2. Telophase 2 में meiosis end with telophase 2 in which the two groups of chromosomes, one again get enclosed by nuclear envelope. Cytokinesis follows, resulting in formation of a tetrad of cells, that is 4 haploid daughter cells. 4 haploid daughter cells. Now what's the significance of meiosis? The significance of meiosis can be understood by the mechanism by which the conservation of specific chromosome number of each species is achieved across the generation in sexually reproducing organisms. Even though the processes paradoxically results in the reduction of chromosome number by half, It also increases the genetic variability in the population of organisms from one generation to the next. Variations are very important for the process of evolution. So it helps in evolution. जेनेटिक वेरिएबिलिटी कॉज करता है और इवोल्यूशन में हेल्प करता है. ये शो कर रहा है डायग्राम स्टेजेस ऑफ मियोसिस वन. ये प्रोफेज वन है, ये मेटाफेज वन है, ये एनाफेज वन है, ये टीलोफेज है, ये भी टीलोफेज है. दिस डायग्राम इज शोइंग द स्टेजेस ऑफ मियोसिस टू. ये प्रोफेज टू, मेटाफेज टू, एनाफेज टू, टीलोफेज टू. Summary. According to the cell theory, cell arises from the pre-existing cells. The process by which this occurs is called cell division. Any sexually reproducing organism starts its life cycle from single-layered zygote. Cell division does not stop with the formation of mature organisms but continues throughout its life cycle. The stages through which a cell passes from one division to the next is called the cell cycle. Cell cycle is divided into two phases. Phase 1, which is interphase, a period of preparation for cell division, and second, mitosis or M phase, the actual period of cell division. Interphase is further subdivided into G1, S, G2, and G1 phase is the period when the cell grows and carries out normal metabolism. Most of the organelle duplication also occurs during the G1 phase. During the G1 phase. S phase marks the phase of DNA replication and chromosomal duplication. G2 phase is the period of cytoplasmic growth. Mitosis is also divided into four stages, namely prophase, metaphase, anaphase, telophase. Chromosomal condensation occurs during prophase. Simultaneously, the centrioles move towards the opposite poles. The nuclear envelope and the nucleolus disappears and the spindle fiber starts appearing. Metaphase is marked by the alignment of chromosomes at the equatorial plates. During anaphase, the centromeres divide and chromatids start moving towards the opposite poles. Once the chromatids reach the two poles, the chromosomal elongation starts. Nucleolus and the nuclear membrane reappears. The stage is called telophase. Nucleolar division is then followed by the cytoplasmic division, which is called cytokinesis. Mitosis, thus, is the equational division in which the chromosome number of parent is conserved in the daughter cell. In contrast to mitosis, meiosis occurs in the diploid cells, which are destined to form gametes. It is called the reductional division since it reduces the chromosome number by half, making the gametes. In sexual reproduction, when the two gametes fuse, the chromosome number is restored to the value in the parent. Meiosis is divided into two phases, meiosis 1 and meiosis 2. In the first meiotic division of homologous chromosomes, pair to both to form bivalents and undergo crossing over. Meiosis 1 has longer prophase, which is divided further into 5 phases. These are leptotene, zygotene, pachytene, diplotene, and dikinesis. During metaphase 1, the bivalents arrange on the equatorial plates. This is followed by anaphase 1, in which homologous chromosomes move towards the opposite poles with both their chromatids. Each pole receives half the number of chromosomes of the parent cell. In telophase 1, the nucleus membrane and the nucleolus reappears. Meiosis 2 is similar to mitosis. During anaphase 2, the sister chromatids separate. Thus, at the end of meiosis, 4 haploid daughter cells are formed.